Hi, everyone. Thank you for all of you that stayed with us in this long day. I want to present now Professor David Olson, which is a chemical neuroscientist with expertise in central nervous system, medicinal chemistry, neurobiology, and behavioral neuropharmacology. He received his PhD in chemistry from Stanford University and is a postdoctoral training in neuroscience at the Broad Institute of MIT and Harvard. Professor Olson joined the faculty of UC Davis in 2015, where he established a research program at the interface of chemistry and neuroscience. His research focused on understanding how small molecules influence neural plasticity, learning, and memory, and mood. In 2019, he co-founded Delix Therapeutics, a company developing non-hallucinogenic analogs. Please welcome Dr. Olson, and thank you for joining us, Professor.
Hi, everyone. Thanks for the introduction. It's nice to be with you all virtually today. So before we get started, I need to mention that in addition to my academic appointments at the University of California, Davis, I'm also a co-founder and the Chief Innovation Officer of Delix Therapeutics. But all the work I'm going to tell you about today was work done in my academic laboratory at the Institute for Psychedelics and Neurotherapeutics at UC Davis. So our group is really interested in developing new treatments for a wide variety of brain conditions. But today I'm going to focus on the so-called stress-related neuropsychiatric diseases. These are illnesses like depression, substance use disorder, and post-traumatic stress disorder. And a hallmark of all of these illnesses is really the atrophy of neurons in a part of the brain called the prefrontal cortex. Now, the prefrontal cortex is the so-called executive control center of the brain. It projects to a whole bunch of other brain regions that do things like regulate mood, motivation, fear, reward, cognition, etc. And so it's perhaps unsurprising that synaptic dysfunction in the PFC can lead to all of the symptoms that we commonly associate with neuropsychiatric conditions. Now, unfortunately, traditional approaches to treat these illnesses have really relied on trying to rectify chemical imbalances in the brain through the use of things like selective serotonin reuptake inhibitors. And these medicines, while they have helped some people, are largely ineffective. They take weeks to months to demonstrate any kind of therapeutic efficacy, and about a third of all patients will never respond to these types of treatments. And so our approach has been a little different. We have been trying to selectively repair damaged neural circuitry to produce both rapid and sustained therapeutic effects after just one or a few administrations of a drug. And we reasoned that the best way to impact the neural circuit is to physically change the structure of the neurons within those circuits. And so for the purposes of today's talk, I'm going to be focusing on these layer 5 pyramidal neurons in the cortex that tend to atrophy in response to chronic stress. And so we started calling these types of medicines psychoplastogens. These are just chemical stimuli that promote structural neuroplasticity in these key cortical neurons, which includes the growth of dendrites. And if you zoom in on one of these dendrites, you'll see the growth of these little tiny nubs. These are called dendritic spines. Dendritic spines are the sites of excitatory synapses in the brain. So these changes in neuronal structure often manifest as functional changes that you can measure with traditional techniques like electrophysiology. Now, one of the first and the best known psychoplastogens is this molecule here on the left, ketamine. Ketamine is a dissociative anesthetic. It's been used in medicine for many, many years. More recently, it was approved by the FDA for treatment-resistant depression. And what I'm showing you here are some cultured cortical neurons that were treated with ketamine. And you can see that the neurons treated with ketamine have more of these dendritic branches than the neurons treated with the vehicle control. And on the bottom here, you can see these green dots. These are representing synapses. So ketamine promotes both neuritogenesis, but also synaptogenesis in culture. And now what I'm showing you is some work from the late Ron Duman and George Agajanian, where they very beautifully demonstrated that chronic stress can lead to that cortical atrophy I was telling you about, the loss of those dendritic spines, which really manifest as synaptic deficits that you can measure with electrophysiology. But what is really remarkable is that a single administration of ketamine within 24 hours can rapidly regrow those lost dendritic spines in the cortex and reestablish synaptic connectivity, potentially explaining its both rapid and enduring antidepressant-like effects. Now, when we got interested in this type of work, we were looking for drugs that might be have some advantages over ketamine in terms of their efficacy and or safety profiles. And one thing about ketamine that's pretty striking is its lack of circuit level selectivity. Its primary target is the NMDA receptor, and the NMDA receptor is found broadly across the entire brain. So we were looking for molecules that might exhibit more circuit level selectivity. And so the first thing we did is we thought about the genetics of the brain. What receptors are expressed exactly where we want them to be in those layer 5 pyramidal neurons of the cortex. And here what I'm showing you is the expression of this particular receptor. It's a serotonin receptor known as the serotonin or 5-HT2A receptor. And you can see that in the rodent brain, it is highly expressed on these layer 5 pyramidal neurons of the cortex, with more expression in the anterior parts of the brain, less as you move towards the back of the brain, and very little expression in structures like the hippocampus or the striatum. So if you're thinking about trying to modulate the 5-HT2A receptor, one of the first classes of molecules that you think about are the classic serotonergic psychedelics, molecules like LSD and psilocin, which comes from magic mushrooms. Now, one of the very first experiments that we did was we simply cultured cortical neurons, and we treated them with psychedelic drugs and then looked at their morphology. And you don't need any kind of fancy analysis tools for this. You can simply visually inspect the cultures, and they look very different. In the middle, I have vehicle-treated neurons, and on the left and on the right are neurons treated with the psychedelics DMT and LSD. And you can see that the psychedelic-treated neurons have substantially more complex dendritic arbors. And if you zoom in on one of those dendritic branches and do some super resolution microscopy, you'll see that the psychedelic treated neurons also have more of these dendritic spines and these filopodia-like structures. Now, there's many, many ways to quantify changes in structural neuroplasticity. I'm just going to briefly describe one way that we use on occasion. It's called Scholl analysis. The way this works is you simply take an image of a neuron, you draw these concentric circles starting at the soma of increasing radii, and then you simply count the number of times a dendritic branch crosses one of these circles. And you can make these really beautiful Scholl plots like the one on the right here, where on the x-axis are the radii of those concentric circles, on the y-axis are the number of crossings. And then you can see that compared to the vehicle control, the DMT-treated neurons have more complex dendritic arbors. Now, this is one way to represent the data. The other way that we like to represent it is simply looking at the maximum number of crossings. So for something like vehicle, it would be about 7, and for DMT, it would be about 11. So if we look at these Nmax plots, what we can see is that psychedelics from 3 distinct structural classes, ergolines like LSD, tryptamines like DMT, and amphetamines like DOI, they're all really good at promoting the growth of these key cortical neurons. Now, these early experiments that we did in culture were really using treatments for 3 days at a time. And we know that in vivo, that's really not what happens. In vivo, you give a psychedelic, it gets into the brain very rapidly, and then it's cleared from the brain very rapidly. So instead of treating the cultures for 3 days, we started giving them shorter and shorter pulses of stimulation, just treating for 24 hours, then 1 hour, and then just 15 minutes, and then giving them time to grow. And what we found is that whether you treat with ketamine or LSD, even these really, really short stimulation periods, 15 minutes, is sufficient to produce long-lasting changes in neuronal growth. But the key is you have to give the neurons time to grow. If you stimulate for an hour, fix and stain, and look at morphology, you see absolutely no change. But if you stimulate for an hour and give them 3 days to grow, then you see really robust changes in dendritic arbor complexity. Now, to demonstrate this in vivo, we really relied on using a rapidly cleared psychedelic, dimethyltryptamine. Now, the half-life of DMT in a rodent is about 10 minutes. And so for this study, we gave a single IP injection of DMT to rats. We then waited 24 hours, a time point when all of the drug has been cleared from the body. We then sacrificed the animals, looked at neuronal structure via Golgi staining and function via electrophysiology. And we find that even a single dose of DMT long after the drug has been cleared from the body results in this very big increase in dendritic spine density. And these changes in neuronal structure are accompanied by functional effects, including increases in both the amplitude and the frequency of these spontaneous excitatory postsynaptic currents. Now, ever since we published this work way back in 2018, it has been replicated and extended by a number of groups throughout the world, but I want to highlight one study in particular. This is work from Alex Kwan's group, and Alex found that a single dose of psilocybin in both male and female mice could produce a long-lasting increase in dendritic spine density, lasting for at least a month after a single dose. Now, the durability of psychedelic effects is one of the hallmarks of this class of molecules, but the other thing that's really interesting about them is that they seem to work across disease indications. And we really believe that has to do with the fact that they're really targeting this critical hub in the brain, the prefrontal cortex. And we know from a whole bunch of opto and chemogenetic systems neuroscience studies that there are circuits that originate in the PFC and project to other brain regions that do things like control drug-seeking behavior, control the expression of fear, and motivation. So to summarize a lot of work really, really quickly, I'm just going to say that in general, psychoplastogens produce therapeutic behavioral responses in behaviors that are known to involve PFC circuitry, such as the enhancement of fear extinction learning, which is relevant to the treatment of post-traumatic stress disorder, and the production of antidepressant-like effects in an assay like the forced swim test. Now, at this point, we were really interested in trying to understand the mechanism by which psychedelics promote cortical neuron growth. And so we started by doing some selective pharmacology. So first, we took an antagonist of the 5-HT2A receptor. We know that in humans, if you block the 5-HT2A receptor, you block the hallucinogenic effects of these drugs. So we thought this was an obvious place to start. And using ketanserin, we can block the effects of LSD, DMT, and DOI on cortical neuron growth. We next wanted to look at a couple of downstream kinases that had been implicated in ketamine-induced neuroplasticity. One is this one here, TrkB. It's the high affinity receptor for brain-derived neurotrophic factor. And the other one is this protein mTOR, which is really responsible for the production of all of the proteins that are necessary for plasticity, all of the structural proteins that allow neurons to change their shape, and all of the ion channels that allow them to change their synaptic weights. And if you block either TrkB or mTOR, you completely block the effects of psychedelics on cortical neuron growth. Now, we realized that when we were using these antagonists, particularly this 5-HT2A antagonist, it is not a perfectly selective compound. It hits a couple of other receptors as well. So to be absolutely certain that the 5-HT2A receptor was mediating this plasticity-promoting effect of psychedelics, we really relied on genetic models. So here I'm showing you in wild-type animals, the psychedelic 5-methoxy-DMT can produce a very large increase in dendritic spine density, but this effect is completely gone in the 5-HT2A knockout animals. Similarly, in the wild-type animals, you get this really big functional effect on EPSC, frequency and amplitude, but the effect, again, is gone in the knockout animals. So next, we wanted to assess the effects of the 5-HT2A receptor on the therapeutic behavioral effects of psychedelics. And so here we're using a modified version of the traditional forced swim test. And so in this version, we've adjusted it to make it a little more challenging, such that traditional antidepressants tend to not work well in this assay. Only these next generation psychoplastogens like ketamine seem to work well. And so the way we do this is on the first day, we do a pre-test that provides some stress to the animal. The next day, we give a single injection of the compound. We then do an acute behavioral assay just to make sure the compound is getting to the target as we expect it would. And then 24 hours later, after the drug has been cleared from the body, that's when we do our test of efficacy. And so you can see here in the efficacy test that treatment with 5-methoxy-DMT can produce this very profound antidepressant-like response, but that if you pre-treat with ketanserin, the 5-HT2 antagonist, you can completely block that effect. Now, the forced swim test is probably more relevant to motivated behaviors. We also know that anhedonia plays a very important role in depression. So to measure anhedonia in a rodent, we rely on the sucrose preference test. So mice, much like myself, if you give them a choice between plain old water and a sugary drink, they will prefer the sugary drink most of the time. And you can actually measure this preference. Now, after some kind of chronic stressor, in this case, we gave daily administration of chronic corticosterone. After about 10 days of that, you can do the sucrose preference test again, and you can see that in both the wild type animals and the 5-HT2A knockout animals, you see this reduction in sucrose preference, suggesting this anhedonic-like state. And then on day 13, we gave a single administration of psilocybin, come back 24 hours later and measure sucrose preference. And you can see a complete rescue in the wild type animals, but no change in the knockout animals. So at this point, we're feeling really, really confident that the 5-HT2A receptor is mediating the plasticity promoting effects of psychedelics and at least some of their antidepressant behavioral responses. But we had a result that really bothered us. And it was this one. It was the fact that this compound right here, serotonin, could not do the same thing as a psychedelic. And that bothered us because I just spent the last 10 minutes telling you that the serotonin 2A receptor is responsible for psychedelic-induced neuroplasticity. And we were really scratching our heads for a long time about this result until we saw this particular structure-activity relationship study. So all we were doing was we were taking serotonin-like compounds and simply adding methyl groups to the nitrogens, going from 1 to 2 methyl groups. And as we added this extra grease, we saw a really nice increase in dendritic arbor growth. And this is very obvious if you look at the Scholl plots going from tryptamine to N-methyltryptamine to dimethyltryptamine, you see that the peak rises pretty significantly. So the next thing that we did is we wanted to correlate activation of the 5-HT2A receptor with this dendritic cortical neuron growth phenotype. And we saw absolutely no correlation. If anything, we saw a slight negative correlation, but this was non-significant. But then when we tried to correlate the cortical neuron growth phenotype with just the lipophilicity of the drug, how greasy is the drug, we saw a really beautiful correlation where these very polar compounds like serotonin were very bad at promoting cortical neuron growth, but these very lipophilic or non-polar compounds like the psychedelics were very good at promoting cortical neuron growth. And that led us to the hypothesis that maybe the reason that serotonin can't do the same thing as a psychedelic is because maybe it can't access the target. Maybe for the 5-HT2A receptor, this particular G protein coupled receptor doesn't exist on the plasma membrane of cortical neurons. Maybe it exists intracellularly, and serotonin is simply too polar to cross the membrane to access the target, whereas psychedelics can freely diffuse across the lipid bilayer of the cell. And so to answer this question, the very first thing that we did is we simply looked for where the 5-HT2A receptor was located. And if you express the 5-HT2A receptor in a non-neuronal cell, like a HEX cell, you see the typical expression pattern. You see a lot on the cell membrane, a little bit inside, but definitely a lot on the plasma membrane. But when you express it in a neuron, or if you just look endogenously, you see something very, very different. You see that pretty much all of the 5-HT2A receptors are located intracellularly. And we found that they localize very, very well with a marker of the Golgi complex. So other people have observed this as well for many, many decades, actually, but no one really understood the functional consequence of this intracellular localization. So to understand that, we took a chemical neuroscience approach. We took these membrane permeable agonists in green and this antagonist, ketanserin, excuse me, and we converted them into membrane impermeable compounds. By adding a methyl group to DMT or a phosphate group to psilocin, we can convert them into the membrane impermeable TMT and psilocybin. Or if we added a methyl group here, we can make this methylated ketanserin. And then what we do is we run the assay under 2 different conditions. One is just plain old neurons. The other is with electroporation. So when you electroporate neurons, you can transiently apply a voltage that will open pores in the cell. That allows impermeable compounds to cross the membrane The pores then close and the impermeable compounds get stuck on the inside of the cells. And when you do that, you see something very interesting. So without electroporation, you get the results that you would expect. You see that the membrane permeable agonists, DMT and psilocin, both promote growth. The impermeable agonists, TMT and psilocybin, do not promote growth. The permeable antagonist, ketanserin, can block DMT-induced growth, but the impermeable antagonist, methylated ketanserin, cannot. But when you electroporate, you get the exact opposite result. Now, the impermeable agonist, TMT and psilocybin, can both promote neuronal growth, and the impermeable antagonist can also block DMT-induced growth. Now, these are neuritogenesis studies. We did the same thing with spinogenesis. And again, permeable compounds promote spine growth, impermeable compounds do not. Now, if this was really true, we reason that if we could just get serotonin to the target, it should be able to do the same thing as a psychedelic. And so for this experiment, we relied on sparsely transfecting neurons with a transporter of serotonin, this protein called SERT. And I love this experiment because it's in the same culture, two neurons side by side. So here is an example of two neurons in the same culture. The one on the left expresses the transporter, the one on the right does not. Similarly, on this side, we see one neuron on the right expresses the transporter, this one on the left does not. Then we can treat the cells with serotonin and we can compare the genotypes. So you see that the vehicle does nothing to either genotype. DMT promotes growth in both genotypes, presumably because DMT can just passively cross the membrane and access the target. But serotonin can only promote growth in the neurons capable of importing serotonin through the transporter. So we then kind of replicated and extended this study. And so here, what I'm showing you, this bar right here is just the replication of that last study that I showed you. So we've got serotonin in SERT positive neurons. But then we tried to block the effect. So we wanted to block it with the 5-HT2 antagonist ketanserin. And you can see right here that it does block the effect. But then we also wanted to block the effect with citalopram, an inhibitor of the serotonin transporter. And if you inhibit the transporter, you also block the effect, suggesting that in order to promote cortical neuron growth, serotonin must be imported by the transporter and then activate the 5-HT2A receptor. You get something very different when you just switch serotonin to dimethyltryptamine. Now, again, DMT promotes growth in both genotypes. Ketanserin can block growth in both genotypes, but citalopram cannot block growth because presumably DMT does not require transport to get into the cell to access the target. These were neuritogenesis assays. We also did this with spinogenesis. Again, DMT promotes growth in both genotypes. Serotonin only promotes growth in neurons that can import the monoamine. Now, to demonstrate this in vivo, we relied on a viral strategy. Now, most of the serotonin transporter in the brain is expressed on the presynaptic side of serotonergic neurons. So normally, serotonin neurons spit out serotonin into the synaptic cleft, and then SERT sucks it back up. SERT is not really expressed on the postsynaptic side, on those cortical neurons that atrophy in response to chronic stress. So we used a virus to express them on those cortical neurons. We then give this serotonin-releasing agent, parachloroamphetamine, come back 24 hours later and look at dendritic spine growth. And what we find is that in neurons capable of sucking up serotonin and importing serotonin on the postsynaptic side, we can get an increase in dendritic spine density, just like we get with psychedelics. Now, if spine growth is responsible for the antidepressant effects, we should see comparable behavioral effects in this type of assay. So we did basically the same thing. We used an AAV to express SERT on cortical neurons. We then did a novelty-induced locomotion assay just to demonstrate there was no difference between the genotypes. There wasn't. We then do that pre-test for the forced swim that I told you about before. Again, you see no difference between the genotypes. We give a single dose of parachloroamphetamine, and we find that the efficacy 24 hours later, we only get an antidepressant effect in animals that have SERT on the postsynaptic side that are capable of importing serotonin. So at this point, we feel very confident that the 5-HT2A receptor at an intracellular location is responsible for the neuroplasticity promoting effects of psychedelics, but also some of their antidepressant behavioral effects. But there is a bit of a problem. Now, I believe that psychedelics are going to be very useful for some patient populations, absolutely. But they were never really optimized to be CNS therapeutics. And as a result, they all have different issues associated with them, depending on their particular chemical scaffold. But one of the big issues related to the clinical translatability or scalability is really the fact that in order to administer them safely and effectively, they have to be given under the supervision of usually multiple healthcare professionals. And that dramatically increases the cost and the complexity of the treatment. And it limits treatment to patients who have a co-occurring condition or a family history of a disorder like schizophrenia, or that might have a personal reason to not want to use psychedelic drugs. And so in order to broaden access to this class of medicines, we wondered if it would be possible to separate the hallucinogenic effects of psychedelics from their neuroplasticity promoting effects and some of their long-lasting therapeutic behavioral effects. And so in order to do that, we needed a really reliable assay that could predict hallucinogenic potential in people. And here I'm showing you the mouse head twitch response assay. It's the behavior here shown on the lower left-hand corner. It's this rapid rotational movement of the head. And Adam Halberstadt's group has done some really beautiful work demonstrating over now probably close to 100 different structurally distinct psychedelic compounds. There is a very nice correlation between potency in the mouse head twitch response assay and human hallucinogenic potential. So we decided to take a psychedelic and try to tweak its chemical structure in the smallest way possible to render it non-hallucinogenic, but also retain the plasticity-promoting effects of the drugs. And so the first thing that we did is we turned to these iso-DMT. So basically all we're doing is flipping this indole upside down. If you had molecular models of these two compounds, I don't care if you're the world's best chemist, you can't tell them apart. They are structurally, equivalent in terms of their spatial orientation and geometry. The only real difference is that the iso-DMTs lack the ability to engage in a hydrogen bond donor like the psychedelics can. And so I'm just going to show you these two structurally related compounds, 5-methoxy-DMT, a psychedelic, and this compound, 6-methoxy-iso-DMT. You can see that in terms of their ability to promote cortical neuron growth, they are equivalent and they're very similar to the potency of ketamine. But when you go in vivo and you look at their hallucinogenic potential, you get something totally different. 5-MeO-DMT causes this nice dose-dependent increase in head twitches. You don't see that with the iso-DMT analog. So the head twitch response is really valuable for predicting hallucinogenic potential in people. But if you want to do drug discovery, you can't really be relying on these types of in vivo assays to screen thousands and thousands of compounds. So we realized that we needed a cellular assay that could predict hallucinogenic potential. And all of the traditional assays of GPCR function, of 5-HT2A receptor function, they really couldn't do this. All of the non-hallucinogenic compounds look like partial agonists in those assays like psychedelics. And so we reasoned that there must be something different about the way that a hallucinogenic and a non-hallucinogenic agonist change the conformation of the receptor. And so the way we solved this problem was very, very simple. We took the 5-HT2A receptor, we just lopped off the third intracellular loop of this GPCR, and we fused on a circularly permuted green fluorescent protein. Now, if you're not familiar with CpGFP technology, the way it works is there's basically a split in the beta barrel that protects the fluorophore, and that allows water to get in, the fluorescence to be quenched. But when a ligand binds to the receptor, that induces a conformational change that closes the beta barrel, and that turns on fluorescence. And so here you can see the agonist serotonin can turn on fluorescence very nicely, but the antagonists, ketanserin and volaneanserin, either do nothing or they turn off the biosensor slightly. Now, we got really excited when we found this result. We found that the potency in this biosensor assay correlated very nicely with human hallucinogenic potency. So we started calling this biosensor cyclite. Now, one of the first experiments we did is we used a virus to express cyclite in the prefrontal cortex. We then gave a single administration of the psychedelic 5-methoxy-DMT, and we see that cyclite is rapidly activated on the same time course as the head twitch response starts to turn on, but it remains activated for a long time, even when the head twitch response starts to wane. If you give the vehicle, you see no change in fluorescence, and if you give the antagonist, you actually see a decrease in cyclite fluorescence. Now, probably the most exciting day in lab was when we got this result, though. And this was when we realized that we could use cyclite to differentiate between a hallucinogenic compound and a very closely related structural congener that was non-hallucinogenic. So here is LSD in green, and this compound, glyceride, is a non-hallucinogenic analog. You don't have to be a chemist to appreciate how similar these compounds are in structure. The only difference is you insert this nitrogen and swap the stereochemistry at that center to make glyceride. Now, LSD turns on the biosensor very nicely. Glyceride does not. Now, you might be asking yourself, well, maybe glyceride just doesn't bind to the biosensor. So to address that question, we just run the assay in antagonist mode. We give a little bit of agonist to turn on the sensor, and then we titrate in increasing quantities of the non-hallucinogenic ligand, and we can watch the fluorescence decrease. So what we've done over many, many compounds now is we've tested them in both agonist and antagonist mode. I want to draw your eye to this black dot here. This is the vehicle control. And so here on the x-axis is agonist mode. So compounds that increase cyclite fluorescence more than 1 standard deviation from the vehicle in agonist mode. These are all these known hallucinogenic compounds in red. And compounds that decrease cyclite fluorescence more than 1 standard deviation in antagonist mode, these are these known non-hallucinogenic ligands in blue. And you see some really interesting things when you look at these compounds in blue. You'll see traditional antagonists like volanerancerin and ketanserin. You'll also see antipsychotics like haloperidol, but you'll also see these non-hallucinogenic agonists like glyceride. And now I'm going to spend a minute to tell you a little bit about this compound here that we call AAZ. It is a structural analog of 5-methoxy-DMT. You have to squint your eyes to kind of see it. We kind of shifted some atoms around a little bit. But the biosensor was predicting that AAZ was going to be non-hallucinogenic. And even when we go up to these massive 100 mg per kg doses, we don't see any head twitch response at all. Now, a therapeutic dose of this compound is probably closer to about 10 mg per kg. So the next thing we did is we wanted to test its psychoplastigenic effects, and we found it had comparable potency to ketamine. And then we wanted to look at its antidepressant-like responses. So again, this is that version of the forced swim test that I was talking about before. It's a little different this time. We did a pretest. The next day we gave the drugs, and then we looked 30 minutes after the drugs just to see if there was a rapid acting effect like ketamine, and we see the rapid acting effect. And then 24 hours later, we come back and we see that that effect is sustained. Now, we also wanted to look at a genetic model of depression. And so for that, we turned to these VMAT2 heterozygous mice developed by Bill Wetzel's group at Duke. We know that in humans, if you inhibit VMAT2, you can induce depressive phenotypes. And these mice have a whole bunch of just baseline depressive phenotypes. But here I'm showing you the sucrose preference test. You can see that compared to the wild type animals, the VMAT2 heterozygous animals have this anhedonia-like phenotype, but a single dose of AAZ rapidly rescues the anhedonia phenotype, and the effect persists. It lasts for at least a couple of weeks before the genotypes start to diverge. So with the advent of tools like cyclite, and some of our structural neuroplasticity assays, we've been doing drug discovery on every single major psychedelic scaffold. Ergolines like LSD, tryptamines like psilocybin, amphetamines like MDMA, et cetera, et cetera. But now I want to tell you a little bit about our efforts to make a better version of this molecule here, ibogaine. Ibogaine is an atypical psychedelic, but it has some pretty profound properties. There are some anecdotal reports and some open label clinical trials that suggest that a single dose of ibogaine can reduce drug-seeking behavior in patients with heroin use disorder for up to 6 months. And a second dose can keep them drug-free for up to 3 years. And these types of long-lasting effects really just scream neuroplasticity to me. So we're very interested in trying to develop drugs like ibogaine. Now, you might be asking yourself, well, if ibogaine is so efficacious, why isn't it used in the clinic very frequently? And there's a variety of reasons for that. Quite frankly, ibogaine is a terrible, terrible drug for many reasons. Number one, it's very hard to synthesize. There are no enantioselective de novo total syntheses that can produce it in the quantities necessary for clinical trials. It is completely insoluble in water, and anyone who's worked with it knows what a pain it is to work with. Of course, it has these hallucinogenic effects that can limit its clinical scalability, but the worst thing about ibogaine is the fact that it binds to hERG channels in the heart and can cause cardiac arrhythmias. And unfortunately, several people have actually died when they've taken ibogaine to try to treat their substance use disorder. So our approach to this problem is really related to this concept called function-oriented synthesis. It's really pioneered by Paul Wender at Stanford, and the idea is really simple. The idea is, can you take a complex natural product scaffold like ibogaine and distill it down into the essential structural features that give rise to the function that you care about? In our case, it would be this neuroplasticity-promoting phenotype. So if you look at the structure of ibogaine, you can see there are kind of some key chemical motifs. There's this indole group here in blue, there's this 7-member tetrahydroazepine, and there's this bicyclic isoquinoline. And so all we did is we simply started to delete bonds. If you delete the bond between the indole and the isoquinoline, you start to make compounds that look like scaffold 1. If you delete the isoquinoline altogether, then you start to make compounds that look like scaffold 2. And I'm not going to take you through all of the medicinal chemistry. I'm just going to show you a couple of highlights. The first thing I want to show you is that, in general, we found that compounds that retained the azepine but lacked the isoquinoline, well, they were pretty good at promoting cortical neuron growth for the most part. But the opposite wasn't true. Compounds that retained the isoquinoline but lacked the 7-member tetrahydroazepine, well, they were pretty bad at promoting growth. And so therefore, we decided to prioritize this compound for further development. We started calling it ibogainolog or IBG because it looked exactly like ibogaine, except it was just lacking that bicyclic isoquinoline. We made one other small structural change. We moved the methoxy group from the 5 position to the 6 position to make this compound. We call this TBG, which is short for tabernanthalog, because tabernanthalog resembles the natural product tabernanthine. Unlike ibogaine, TBG is much easier to synthesize. We can produce it in just a single step in gram quantities through this Fischer-Indole cyclization. It is also very, very water soluble, and it is a potent partial agonist of the 5-HT2A receptor. Now, I want to show you this one structure-activity relationship study because I just think it's really interesting. You'll all remember that the structure of 5-methoxy-dimethyltryptamine is embedded within the core scaffold of ibogaine, and 5-MeO-DMT produces many of these head twitches. But if you just conformationally restrict the dimethyl amino group to the 2 position of the indole, you can produce this compound IBG, which has a lower head twitch response. And then by moving the methoxy group 1 position over, now you've completely eliminated the head twitch response. Moreover, you've not just eliminated the head twitch response, you produce an antipsychotic-like compound. Because if you pre-treat the animals with either vehicle or tabernanthalog and then give them 5-methoxy-DMT, you can find that TBG can block the psychedelic-induced head twitch response. So now we've got a non-hallucinogenic variant of ibogaine. What about the cardiotoxicity profile? It turns out that by cutting all of that extra grease off of ibogaine, we've dramatically reduced the potency of TBG for hERG. So here's the potency of ibogaine. Here's the potency of TBG. You can see it's about 150-fold less potent. And this is so profound that you can actually see this in the heartbeats of these immobilized zebrafish. Here's the vehicle-treated zebrafish heart rate. You can see the bradycardia, the slowing of the heart with ibogaine. And you can also see that on the bottom there, that TBG's heart rate looks just like the vehicle control. Now, in addition to the cardiotoxicity effects, ibogaine just produces some general toxicity issues. You can look at these pie charts here on the left, but I think a picture is worth 1,000 words. This is what a vehicle-treated fish looks like. This is what an ibogaine-treated fish looks like. They are very, very different. You can see that a tabernanthalog-treated fish looks much more similar to the vehicle control. Now, It's great that we produce a safer variant of ibogaine, but none of this matters if it doesn't have ibogaine-like efficacy. So the first thing that we did is we assessed the psychoplastogenic effects of TBG in vitro and compared it to ibogaine, and they are very comparable. But here is an in vivo study. So for this study, we took these mice that have a subset of their cortical neurons labeled with a fluorescent protein so that we can visualize them. We then cut a window into the skull, and we use a 2-photon microscope to look at a very specific section of dendrite. We then give a dose of TBG, come back 24 hours later after the drug has been cleared from the body, and we look at the exact same section of dendrite. And what that allows us to do is to quantify how many dendritic spines were formed, like this one here in blue, and how many spines were eliminated, like this one here in red. And what we find is that TBG does not impact spine elimination, but it does promote spine growth, and it does so to a comparable extent as the hallucinogenic drug DOI. So again, if these changes in dendritic spine density are therapeutically relevant, we should see broad therapeutic effects across multiple behaviors known to involve PFC circuitry. So the first thing that we did is we looked for antidepressant effects in the forced swim test, and we found comparable effects to ketamine. After that, we worked with Dorit Ron's group to do some studies in these mice that are exposed to this 2-bottle choice paradigm of alcohol consumption. So basically, these mice start to exhibit binge drinking-like behavior. We then give a single dose of TBG, and we find that pretty much every single animal has a reduction in their alcohol consumption without any impact on their total fluid or water consumption. And then finally, we looked at a model of heroin use disorder with Jamie Peters. This is an assay where these rats are trained to lever press to receive an infusion of heroin. And we pair that infusion with a light and a tone cue. And then we can reestablish drug-seeking behavior by playing the tone and showing them the light cue. And what we find is it doesn't really matter when we administer tabernanthalog. We could administer it during self-administration or extinction training, or immediately prior to cued reinstatement. But in all of these cases, we produce this long-lasting protective effect on heroin-seeking behavior. And, during self-administration, this is several weeks before we reinstated the behavior with the drug cues. And so finally, I just want to mention that tabernanthalog has the potential to heal damaged neural circuits. So this was a study we did in collaboration with Yi Zhuo's group, where we provided unpredictable mild stress to animals. And what you can see here is that in the somatosensory cortex, when mice whisk and they like to explore their environment with their whiskers, you see this really nice coordinated calcium response. But unpredictable mild stress completely disrupts this coordinated response. But what is really remarkable is that a single dose of tabernanthalog after the drug has been eliminated from the body can restore this coordinated response, suggesting that these types of psychoplastogens or non-hallucinogenic psychoplastogens can actually repair damaged neural circuitry. So to summarize what I told you about today, I told you that in a healthy brain, neurons in the prefrontal cortex have lots of dendrites, and these dendrites are studded with lots of these dendritic spines, which are the sites of excitatory synapses, but that in many, many brain conditions, and today we focused on neuropsychiatric conditions, but also neurodegenerative conditions, there's an atrophy of neurons in the prefrontal cortex, and psychoplastogens are particularly good at rescuing this cortical atrophy. So in summary, I told you that psychedelics promote structural neuroplasticity by engaging intracellular population of 5-HT2A receptors. I told you that this biosensor Psychlite is pretty good at predicting hallucinogenic potential in vitro, and that TBG and AAZ are potentially two safer psychoplastogens that might be more clinically scalable than first-generation psychedelic compounds. And with that, I'd like to thank these funding agencies on the right, as well as these key collaborators here. And finally, I need to thank all of the really incredible students I get to work with every day. They're super hardworking, very creative, and just kind of a joy to work with. And so Calvin was the first author of the Cell Reports paper. Lindsay was the first author of the Tabernantholog paper with some help from RJ. Max was the first author of the science paper about intracellular 5-HT2A receptors. Lee and Calvin were co-first authors along with a student from Lin Tian's group on the PsychLite paper. And with that, I'd just like to thank you for your attention, and I'd be happy to answer any questions.
I will stand here that you will be able to see me too. Thank you very much for amazing talk. I don't know how many people are here really scientists and can understand the depth on and the breaking through systems and methods that you used in this work. Just caused me to envy, not more than that. Questions? Yes, please.
Hi, thank you for that talk. That was super interesting. I wanted to ask a question about, so you demonstrated how the more hydrophilic compounds like serotonin and NMT don't exhibit the neurogenesis effect as strongly as the hydrophobic compounds because they don't cross the neural cell membrane as well. But in terms of the in vivo results in the forced swim test in mice, have you considered that might also, at least in part, be due to a pharmacokinetic effect of their inability to cross the blood-brain barrier?
Oh, absolutely, 100%. And that's kind of tied to the inability to passively cross membranes, is intimately tied to pharmacokinetic effects if you're administering a compound systemically. This is one of the reasons the body likes compounds like serotonin as neuromodulators, is because most of the serotonin in your body is actually produced in your gut, and it doesn't actually passively diffuse into the brain. So the serotonin that's in the brain is really coming from neurons that produce it in the brain.
Thank you. Any other questions? So I have one very biologic one. You showed that the 5-HT are mostly in the Golgi, in the neurons. And I'm wondering whether this is a way of regulation of the cells when to express them on the membrane, like, you know, the Golgi is the post office of the cell, to send them to the membrane, or it's really functioning in the Golgi. And if it's functioning on the Golgi, what does it doing there? I don't find the logic about that.
Yeah, it's a really important question. And we've been trying to dig into this for some time, and I can give you a few hypotheses. So one hypothesis is that this Golgi population is kind of laying in wait. It's kind of like an inactive population that is waiting for the right stimulus to then go to the plasma membrane and then be stimulated by serotonin, an endogenous ligand. That's one possibility. We looked to see if psychedelics cause pharmacochaperoning. So like you give the drug, does more of it get to the plasma membrane? We didn't find any evidence of that. But it's still a possibility. The other possibility is that there might be other genetic mechanisms that can regulate the ability of things to activate that intracellular population. Remember when I told you that we express the serotonin transporter on those postsynaptic neurons, and then serotonin can activate them? Well, it turns out that the body regulates the expression of that transporter too. And it seems to be the most highly expressed on those postsynaptic cortical neurons during the periods of highest neuroplasticity during development. So it could be that the body is using this mechanism to turn on neuroplasticity early on in life when we need to wire up the brain, and then it shuts it off later in life in favor of neuronal stability. But the final hypothesis, and my favorite of the three, is that serotonin is not necessarily the only molecule that could activate this receptor and not the endogenous ligand for this intracellular population. And that leads us to the question of, does the body produce endogenous psychedelics? And my group has been working on this for a long time, as has many other groups. And I think the answer is The bodies unequivocally do produce psychedelics. All of us have psychedelics in our bodies. The big question is, when and where are these psychedelics produced, and are they produced in the quantities that are sufficient to cause some kind of meaningful physiologic response? And that is questions we're still working on.
I will just say that we know that endocannabinoids are binding the 5-HTA. So that's one example of molecules that are not serotonin that affecting that. I'm wondering if this is also the answer in the gut, because we know that it's changing the expression of the cert in this different stimulation. So maybe it's also working there too. We will check that. Bernard.
Thank you very much, David, for a fantastic talk. You showed data from Alex Kwan, and certainly the other data you showed and the clinical effects suggest that the effects of neuroplasticity of psychedelics and of the new compounds you have synthesized are extraordinarily long-lasting. A single dose can go up to a month, maybe longer. I wonder if you could speculate in what direction, how you are thinking. How does this come to pass? I mean, this is not a trivial thing, that a single dose of a drug can be sufficient to induce neuroplastic effects which extend at least a month and maybe even longer. There has to be something really remarkable going on.
Yeah, it's an important question. And so I'm going to answer this question with an analogy. And so no one would question a treatment response for someone who broke their leg. If you break your leg, you go into a cast. Once the bone is healed, you can take off the cast and your leg is healed indefinitely. I do not think that it's that much different with these agents. I think that they are really inducing the repair of the damaged neural circuits. And once the repair happens, they can have long-lasting effects, as long as they're not re-damaged, as long as there's not an insult that continues to damage those circuits. And we've actually done some imaging in vivo over time to look at this. And this is an area where our work is often misinterpreted. People think that when you give a lot of these drugs, you're just producing rampant neuroplasticity all over the brain indiscriminately. That's not true. It turns out that when you look in an animal before depression, you can induce depression with chronic stress, and you can see a reduction in dendritic spine density. And then when you give one of these agents, you see a rescue in dendritic spine density, but it's not just the total dendritic spine density. We can look at the synapses that were lost, and more often than not, it's the exact same synapse that was lost that is reconstituted after the drug is given. And we think that has to do with the fact that the dendritic side, the dendritic spine side, the atrophy happens more easily, but the presynaptic axonal bouton more or less remains intact. And so when you give the drug and you start promoting growth, more likely than not, you're going to link up the damaged synapse. So you're just restoring the synapses that were there prior to the insult. And that's why we think they have such long-lasting therapeutic effects.
Conceptually, that is a really important point that you are making. Most people do think, as you indicated, that enhanced neuroplasticity means that the neuroplasticity is going on all the time. And what you are postulating is actually a repair of damaged neuroplasticity, which then remains repaired. So you would not find enhanced neuroplasticity. So then how come you see enhanced neuroplasticity a month later? You're not seeing Alex Kwan is not seeing a correction. He's actually seeing enhanced. The mice that he's examining with his methodology are not mice that have been subjected to a chronic mild stress or something like that.
Yeah, so the answer to that is, and we do it too, the answer to that is the dose. So in a wild type animal, in order to get any kind of growth, you have to use pretty heroic doses. And that is because there are homeostatic mechanisms in the brain that prevent overexcitation. The brain doesn't want to have epilepsy or something like that. So if you start producing too many dendritic spines or too much excitatory neurotransmission, the brain naturally culls them back. And so we found that even with tabernanthalog, in order to get a response in vivo, we need to use 50 mg per kg of a dose, which is a pretty big dose. We can get a wild type animal to have an increase in neuroplasticity if you give a high dose. But if you induce a deficit through chronic, unpredictable, mild stress, now you have to use a significantly lower dose to rescue the effect. You only have to go, 10 mg per kg kind of max at that point. And so I think the way they're going to be used in the clinic is not really to promote a lot of extra growth in people, in healthy individuals. It's really going to be to repair the damage in patients.
Thank you so much. I was curious, you mentioned the antipsychotic effects of TBG, and I'm wondering if you think there might be a clinical or therapeutic application for psychotic spectrum disorders and maybe even dementia or other cognitive decline?
Yes and yes. And so, and without getting too much into this is actually an active area of research for my company, Delix Therapeutics, but it's also something we're doing on the academic side of things. I got really excited about this area in particular because of schizophrenia. It turns out that one of the most replicable findings in schizophrenia is atrophy of cortical neurons. And it's believed that perhaps the atrophy of those neurons is really responsible for some of the negative and cognitive symptoms of that disease, for which we don't really have great treatments. We have really good treatments for addressing the positive symptoms of the disease, but not the negative and cognitive. And so we've actually been developing analogs of psychedelics that kind of do both. They promote cortical neuron growth, but they also act as 5-HT2A, partial agonist, antagonist that can kind of block hyperserotonergic transmission, just like a typical antipsychotic like clozapine or pimavanserin or something like that. And so this is an area where these non-hallucinogenic psychoplastogens, I think, have substantial advantages over a compound like psychedelic. You're not going to want to administer a psychedelic to a patient with psychosis. On the neurodegeneration front, I also think this is a similar argument. It's very difficult to give a psychedelic to a patient with dementia. It's hard to manage the kind of anxiogenic effects that those compounds might have and the expectations of that patient. But the non-hallucinogenic psychoplastogens could be more safely administered, and they have the potential to actually restore cognitive function. They're not just halting disease progression. If you can catch it before the neurons actually die, you might actually restore some of the synaptic loss. And we know that in diseases like Alzheimer's disease, synaptic loss correlates best with the cognitive impairments of the disease.
Thank you for the talk. So I have a question. You have shown that some modified molecules, like you don't have the head twitch anymore with the mice, but you still keep some of the neuroplastic effect, and that these neuroplastic effects still correlates with the behavioral effect, right? So do you believe that the head twitch or the psychedelic effect on the mouse also contributing something to the behavioral effects, or the only thing that contributing is the plasticity, the enhanced plasticity?
It might depend on the behavior, but I can say that for some of the antidepressant-like effects that we see in assays like the forced swim test or the tau suspension test, we think that the long-lasting effects are primarily driven by the dendritic spine density. And the reason we know this, and this is unpublished right now, is we've done some studies with Connor Liston where we've photoablated psychoplastogen-induced dendritic spines. And when you do that, you lose the long-lasting antidepressant effects. He's done this with ketamine. He published a really beautiful science paper in 2019 demonstrating that spine growth was causally related to the sustained antidepressant effects of ketamine. And we see something very similar with the other psychoplastogens as well.
Okay, David, thank you very much for staying us and answering all these questions. Amazing talk, one of the best that I heard in the last year. So thank you very much for being with us, and thank you all. Thank you
Thank
very
you all.
much.
Bye-bye.